Hi, everyone. As part of this comparative genomics section, we're going to talk about taking a genomic region and blasting that and how you confirm things. So let's just give you the setup here. Using the protein family sorter and the anchoring function, we found a region in Brucella microti and some other genomes that was uniquely shared to them, and some genomes are missing in it. Most notably, the Brucella abortus genomes. And these are just a subset of 39 genomes from Brucella abortus, and abortus has probably hundreds of genomes in PATRIC. So it's just a subset of that. But this one particular genome, abortus biovar 5 strain 3196, which you can see in the pop-up window there, it has some of the proteins in this region. But for the most part, it looks like Brucella abortus is missing it. So we had drawn a picture over, we'd drawn with our cursor a region over that corresponded to that genomic island. That generated a feature table that showed what those genes were. We chose the middle gene, which is this TRA-I gene, and looked at that gene. And then in the last video we did, we went to the genome browser and we learned how to adapt the genome browser, change the sequence to map the region. We went back into the table and added the start and stop, and then we adjusted it here. And then we saw that if you click on the reference sequence, save track data, and visible region, and view that, you can capture the data that the nucleotide sequence that corresponds to that genomic island. So let's copy that. And let's go to a different PATRIC page. I'm just going to overwrite it here and go up into services and click blast. Let's paste the sequence here. And PATRIC immediately recognizes what it is. And it says, do you want to search against the nucleotide database or the protein? Well, we're looking for this genomic island, so we want the nucleotide. Okay, and the default is set for reference and representative genomes. But remember, we're asking, is it true that Brucella abortus is missing this with the exception of some pieces here? Let's go back here and click the down arrow box here. And there are a number of things we could blast against, but at the very bottom it says search within a taxon. So let's click on that. And then it wants you to tell what the taxon is. So the taxon I want to verify that it's missing from, or check it out, is see if the heat map is correct, is Brucella abortus. And as I start typing, it starts trying to find something, and up here it says species Brucella abortus. So let's click on that, and we're looking for genomic sequences or contigs, and we're just going to set it very loosely, the e-value threshold, and let's click search. So it's blasting against all the Brucella, and at first you look at this and you think, wow, it's hitting a lot of the Brucella abortus. It must be here. But let's dig into this a little more closely. Here's the genome. Here's the accession. Here is the contig. So this is contig 20. It's 100% identity. So of the query that I gave it, and the query was this sequence, 84% had 100% identity. And that was what does this mean? Subject 7%? Well, remember, it's looking at the whole contig here. So here's the query length of that whole genomic island is 18,964. 84% of that is covered in the blast hit. The subject, the whole contig, is 226,340. So it is only 7%. and there's the score and the e-value. Okay, that looks pretty good. Then we have this particular genome that has 79% query coverage, and it's a smaller, obviously a smaller little contig. You can see how that it's smaller here compared to that one. So it's 82% there, so we've got less. And then we start, oh look, we're getting the same genome here. Dump, bump, bump, bump. This BioVar 5 strain 3196. So it's still getting 100%, but then it's smaller pieces of the query. These are actually not so great. It's 7%, 6%, 6%, 6%. And actually, these are tiny little contigs. So they're very small. So it's just hitting little pieces of this contig. And if we go back to the heat map view and look at this, you can see that it's sort of what we're seeing here, just a few of these genes that it's hitting. And then we go down here and things get even worse. It's even hitting a smaller portion. So if you want to see what's being hit here, we can click this down arrow here, and you can see how much of it is actually being hit in the blast hit. So it's a small little piece, clicking there, as compared to this one, which was a good one. So let's click that open, and you can see that it's a much larger piece. It's starting at this part of the query sequence. So it's that particular location. And so that's a much better hit. So that's a good way to open up the results in a blast sequence. So I would still argue that I would trust this and this, and even these little pieces, even though they're small. But when it gets to this is just terrible. So it's probably just pieces of the chromosome that are conserved across that. It's nothing like what we would expect to see with a good, strong identity, especially if it's only 5%. And you know, to be honest, this isn't so great either. So I would argue that out of all the Brucella abortus, these two are the ones that have the best hit to this, and that everybody else just has tiny little pieces. These guys have got most of it. And if you want to know how many Brucella abortus genomes there are in PATRIC, just going to open up another tab here, and let's go into organisms, and then scroll down to Brucella. And this is all the information, summarizing all the information for all the Brucella genomes. There are 1,053 total genomes in Brucella. How many of those are abortus? Well, we can find that out by clicking on taxonomy. And you see abortus is here at the top. We click on that. And then the vertical green bar tells us we could go to the taxon landing page. So let's click on that. There are 408 Brucella genomes in PATRIC. When we did BLAST, we got hit really good hits to only 2 of them and a partial hit here. And then the rest is kind of, you know, not so great. But these are the 2 best ones. So that's how you can use BLAST, going from a genomic region and seeing how strongly it's conserved, or if you can find it somewhere else. Just for fun, let's click Edit and Resubmit. And scroll down to, we've got to get down to the databases. Just for fun, I don't want to search within a tag, a plasmid, a taxon. Let's see if we have any hits to plasmids, known plasmids. And let's search that. And we'll see what it comes up with. And it comes up with some not great, like 6%. So No, unfortunately, we're not seeing any plasmid hits for this, but that's one of the things that you can use in PATRIC, and one of the ways you can use our different databases to explore different sequences that you're interested in. And so next week, we'll start talking about building code on trees. And thank you for listening, and thank you for using PATRIC.